Fine. So now, what are the examples that this CRISPR screening has been done to identify things? So there is a lot of work going on where people have used this CRISPR screen genome-wide using human guide RNA library to find out, for example, the role of certain genes and certain drugs in cancer metastasis. So there is a lot of cancer research going on. There has been CRISPR screen to find out the cell pathways in cancer that trigger some cell-based detection. They have been also used to identify breast cancer vulnerabilities and even how it interacts with the signaling pathway. And of course, I mean, see, after the SARS-CoV-2 pandemic, the screens have revealed what kind of proteins inside the cell are required for SARS-CoV-2 infection and genome-wide, right? Out of 20,000 odd genes, how many are required for SARS-CoV-2? And that those can be developed as therapeutic drug targets in the future, right? So these are the advantages of using the CRISPR screen, and there is many more for drug and toxin, etc. Lot of things we are understanding the biology of the human cells or even yeast and other cells by using these screens genome wide. Now I will talk about something else that you might have heard, which is called gene drive. So gene drive is also based on CRISPR system. So what happens is that in this gene drive, you have to put a Cas9 guide RNA containing cassette within the genome in such a way that the guide RNA will target the loci right next to it. Because we are diploid, so one of this chromosome has this Cas9 guide RNA cassette, the other chromosome doesn't have. But since you have put a guide RNA and a Cas9 together, it will go and cut. And if it cuts and there is no donor template, this chromosome will use this sister chromatid as a donor and it will copy itself, right? So the repair will take this other this sister chromatid as a template to repair itself. So now what is happening is that when the cell divides, immediately it will have a biallelic situation. So this is a very powerful technique which was sort of accidentally discovered, but now has a lot of potential as well as threats, as we will see and you can think of. So these are called gene drive. And why they are called drives, gene drive? So we know in Mendelian inheritance, if we look at, if we have one mutation in one of the strands, then 50% of the progeny will inherit it. And if you cross that mutant, right, with a wild type again, then again 50% and so on. So whatever modification you want to do will spread in a Mendelian fashion, which will be very slowly, although it will go, but a large amount of this, in this case, an example of mosquito, they will not have the gene that you want. But if we do a gene drive, then I said they can be biallelic. And since it is a CRISPR cut, there is no, kind of a homologous recombination thing. It is cutting, DNA is repaired, and it is taking the other strand. So by that mechanism, if you take the mutant mosquito, cross it with a wild type, then 50% of the next generation, or all the generation will be mutant. If you take again that parent, all the generations. So if you go on crossing with the wild type, within a few generations, the entire population will carry the gene of interest. This you can clearly see is in contrast with the Mendelian inheritance. So this is called sometimes super Mendelian inheritance, or it is called gene drive based inheritance, right? So here you can, that means propagate a given character or a trait in the population very fast. Now, why it is useful and why it is even dangerous, right? So this has been used, or currently planning to be used. I mean, it has been used in laboratory condition, but not in wild, to replace mosquito population. We know there are a lot of diseases caused by mosquitoes, right? Malaria, dengue, and all those kind of things. So vector-borne diseases. So one idea is that can we then introduce one mosquito which will go and mate with the wild type population, and with time the wild type population will crash, the mutant population will pick up, right? And that is a strategy which is called population replacement. That means you can replace a wild type mosquito population with a recombinant that you have engineered. You can also do population suppression. That means if that gene targets a fertility gene which is required for mosquitoes reproduction, then what will happen? It will go and mate, and after some time this entire population will crash to zero, because you have knocked out a gene which is required for laying eggs or some kind of reproductive behavior. So within a few generations, in a very short time, this entire population of the mosquito can be wiped out. And these strategies were used partially during the Zika pandemic in South America and Brazil, that they actually killed off literally all the mosquitoes in a village just to stop the spread of the disease, because these mosquitoes, they carry the virus. So how it has been used, I mean, you can see that there are a lot of potential, there are a lot of discussion going on, research going on, that can we modify genetically modified mosquitoes, we can program it so that they can eradicate malaria, right? They don't allow the malaria parasite to grow within them, or kill off all Anopheles mosquito. That is one way. So you can have an anti-malaria peptide expressed in the gut of the mosquito and let that mosquito go and replace the population. Then there will be no more malaria spread. So it can bite an individual, but it cannot spread the parasite again. There are some concerns because of this. That means, it can be used as a bioweapon. So there are some now self-destructing mosquitoes and some rodents which are now being developed so as to contain in the wild type. In the wild population, if required, we don't want to kill off everyone, but we can also destroy the gene drive. So there are variety of checks and balances going, but the potential of this technology is enormous if used at the right hand at the right place, right? So these gene drives can be used for pest management, even for controlling invasive species. In agriculture, a good trait, let's say salt tolerance or a higher production, this trait can be cloned into a gene drive and spread into the population very quickly without going for traditional breeding. Of course, we can kill off any pathogens and any vectors we want, which are not important to us, and it will help in controlling all these vector-borne diseases. So this has a lot of potential, but also there has big concerns, and there are a lot of checks and balances. So now I will switch gears. We have seen what Cas9 can do, but now, of course, there are many different Cas proteins available. So Cas9 we saw creates a double strand break. There are another class of Cas protein called Cas12, not 9. And what it does, that it creates a break, but what also does, if there is a nucleic acid close to this protein RNA complex, it cleaves that as well. And that is called transcleavage, or it is also called collateral damage. This property of this Cas protein is not only unique to Cas12, but there is another protein called Cas14, which also has this transcleavage. And as you might know, there is another protein called Cas13, which targets not DNA, but RNA. And when this also, so you can cleave RNA by this protein. So once it cleaves the RNA, this protein also has this transcleavage property. So that means it can have so-called off-target effect, because it will indiscriminately cleave nucleic acids around it. But that has been used for good purposes, and that is an upcoming field, that is diagnostics. How this property has been used for diagnostic. So let's assume we have a viral DNA in our blood. We want to check if there is a virus we are infected with. So one can design probes, and these probes can be simple double-stranded DNA with a fluorescent dye attached on one end and a quencher dye attached at the other dye. So when they are together, this quencher will not allow the fluorescent to fluoresce. That means it will be, if you look under a microscope, it will be dark. Now you take a blood sample and you take this Cas12, or in this case Cas12A. You complex it with the guide RNA. This guide RNA should target the virus that you are looking for. Can be dengue, can be AIDS, can be anything, right? And this, when it goes and binds to the viral DNA, will cleave. Once it cleaves, this Cas12 will go crazy. It will start cleaving all other DNA that is around it. And you have soaked this reaction in a tube along with these DNA probes. So normally this DNA probe is dark, but if the Cas has cleaved the virus, and only if it has cleaved the virus, it will start cleaving them also indiscriminately. That will release the quencher from the fluorescent tag, and now you can see fluorescence, right? So that means this indiscriminate cleaving of DNA has been used to cleave a quencher fluorescent pair to make the fluorescent. So this has also been used for viral RNA, like the viruses which have RNA genomes, and you can use Cas13, and you basically end up having a fluorescence. The idea is very simple. That means in the pipeline, you can collect blood samples or whatever samples you want from a patient, isolate DNA and RNA, depending you are looking for DNA virus, RNA virus, or any kind of pathogen, amplify them. You add this Cas12 or Cas13 protein with this, mixed with this a fluorescent tag and a quencher mix, And then because of this collateral activity, this will get cleaved, and you can either read it in a fluorometer, or you can also develop a lateral flow assay, which is a dipstick method. So people have actually, this is not a theory, so people have actually done it, and it was published in a Cell journal, where there is a rapid low cost detection of Zika virus using these kind of synthetic biology approaches. So uh, I don't want to go into the detail, and you don't have to know, just you can appreciate that this diagnostic test from blood sample collection to detection of a virus is less than 3 hours. Currently, if you have a dengue, then you have to go for ELISA or some other thing that takes much more time. But using this technology, yeah, you can do it at room temperature around 20 degrees, and this test can differentiate between different flaviviruses. Dengue, Zika, they belong to the same class called flavivirus, but you can still distinguish between them. And this entire technology is based on CRISPR. How does CRISPR cleave and guide RNA is targeted? So CRISPR-Cas12, of course, has also been used to develop diagnostic test for SARS-CoV-2. Similar manner, you have to take this SARS-CoV-2 N gene, the envelope gene, nucleocapsid, etc., mix it together, single strand probe gets cleaved, and you see a fluorescent or a color. And you can do a dipstick test, right? So that means you have to remember that Cas9 cannot be used because it doesn't have collateral damage property, but the other ones can be used, and they have been used for diagnosis. Okay, I will just skip this. I will just quickly go through the dead Cas system since I have limited time. So this Cas9 protein, we know it goes and cleaves the DNA and puts a double strand break. Now, what people have done, they have just introduced two amino acid changes in this protein, knocking out the nuclease activity. So now this protein Cas9 can bind the DNA but cannot cleave. That means it is a dead protein, and that's why, and so it is called a dcas9. So what you can now do is that you can, let's say, take the dcas9 protein, same guide RNA targeting some gene, but this dead cas9 can be fused to a protein domain which can either activate transcription or repress transcription. That means if you can now target this guide RNA and the dead cas protein in the promoter region, you can artificially turn on or turn off genes, and it has been done for the last 8 years or so. The important point to remember is that there is no change in the DNA sequence, right? Cas9, Cas12, they were all cutting DNA or RNA. In this case, because it is dead, it won't change the DNA sequence. So it is a much safer way to do things. So this has a lot of potential. Again, for the sake of time, I don't want to go in detail, but there are a lot of ways it is currently used. I want to just highlight one latest application of this dead cas system and how it is used to correct diseases. So there is a application which is called base editing. That means you can change the nucleotide of a DNA literally at will. So what you do, you have a dead Cas9, you have a guide RNA, but the dead Cas9 is fused to a deaminase. Now, if it is a cytosine base editor, the deamination of cytosine will lead to formation of uracil, right? Wherever in the DNA you have targeted. And now this uracil cannot base pair with the guanosine, which was coming from the other strand. Because of this mismatch, there will be repair. And the DNA repair machinery will keep this uracil but will remove guanine and put a adenine. So now what we have done is basically you have changed a CG base pair to a AT base pair when the DNA replicates. So this is called base editing. One base change you have done. So you can not only change cytosine, but I'm not going to detail. You can actually change the AT pair to a GC pair. So that means you can do the reverse also. Not only from CG to AT, but AT to GC is also possible. So just by fusing a deaminase protein with the dead Cas. And this has been used for editing in CAR T cells that I was telling you earlier. And this has been used to treat blood cancers. Now, how it is done? Normally, a T cell will have these kind of receptors. Don't have to remember the names, but all you have to know is that this base editing is used to knock out these receptors, leaving a normal T cell without having a T cell receptor or this other co-receptors. Now, what you do? You come out with this synthetic receptor that I was mentioning earlier, and that you infect. I mean, you make these cells express that CAR, right, which is a chimeric antigen receptor in this T cell, and this CAR is against CD7, and the CD7 is present in the leukemia cell, right? And now, if you put this CAR T cell and infuse in the blood, it will go and attack the leukemia cell and it will kill it. Of course, you have to do a bone marrow transplant to put healthy T cell, but at least the damage, the cancerous T cell is taken off. So this is a CAR T cell therapy using CRISPR technology, especially the base editing technology. So this was used for the first time last year, and that was the world's only case where she was terminally ill. So pediatric leukemia is now being treated using this base editing technology, and that was much in the news. And now this base editing is also used to correct cholesterol levels. So that is a major reason for stroke and heart attacks, right? High cholesterol. So what scientists have done, they have found an enzyme called PCSK9. What does this enzyme do normally is that it removes the LDL, which is a bad cholesterol receptor inside the cell. So that leads to a high LDL in the blood. What they have done, they have removed this enzyme. If you remove this enzyme, inactivate by base editing, then this receptor cannot go inside. And if it cannot go inside, it will sequester the LDL in the blood. So you will have a low bad cholesterol in the blood. And that has been done. A company has patented it. Now they are going for clinical trials. But the idea is to have low cholesterol, low bad cholesterol in the blood, so that people will have less of heart attack and strokes. Okay, I probably will go and skip these because of the sake of time. We can have maybe another online talk how to do the livestock. I will just highlight here one thing, that now not only humans, CRISPR is used to edit other animals, and this is for meat purpose, or in this case, they edited the pig genome so that they don't have the pig viruses, so that these organs from the pigs can be used for transplantation in humans, because there is an organ transplantation shortage, and the pigs organs come sort of close to the human organs, but they don't want the viruses in the pig genome to come into the human genome. So now they have made the pigs which are safe for transplantation, and that is useful for xenotransplantation. CRISPR-Cas9 has also been used recently to edit the silkworms to produce spider silk, right? And the spider silk, as you might know, is much stronger than nylon and is tougher than Kevlar. And Kevlar is used for bulletproof textile, right? So this spider silk dresses will come in the market soon, which will basically will be the toughest material, but one of the most softest. It's not like metallic, right? So now we shift to agriculture. We talked about human, we talked about little bit about other animals, but let's go for the plants. So what is there in the agriculture? Especially, we are a country which is having a huge agricultural base, and the farmers are involved. So CRISPR can be used in principle to modify the plant genome to make them more drought resistant, salt tolerant, and change in the climate that can happen. And this has already been the work in progress. They have also been used, for example, to prevent browning of the mushrooms and cut vegetables. Many vegetables, if you cut, there is a phenol oxidation. They turn black or some other color, and that is bad. So you cannot keep it for long, and it loses value. So can we use CRISPR to knock it off? The CRISPR has also now currently being used to prevent the plants or the crops from different pests, bacteria, and viruses. They also have been used as a biofactory to produce vaccines. So the trial Ebola vaccine MZAP is actually produced in the tobacco plants, and it was extracted from there. So they can also be used to make drugs. We know that artemisinin is a drug, but they can be engineered to put in another plant. The CRISPR can also be used to harness more solar energy to convert into more efficiently the biomass, right? So more efficient photosynthesis, for example. They have also been used to increase the nutrient value of different fruits and vegetables. For example, tomato has lycopene. They have edited some other genes so that it becomes much more nutritious. The banana has been actually modified by CRISPR to put carotene. So these bananas, they look a little bit pinkish, but that is healthier food, right? So in addition to the carbohydrates. So there are a bunch of disease resistant strains now getting generated. This is a list of the trait and the genes that has been targeted in different crops, and these all of these crops we eat. I mean the vegetables and the other rice, wheat, etc. So these are very important going forward to make them more insect resistant, to make them more tolerant, to actually have food safety. Cassava has some cyanide, so they're trying to make a cassava which is cyanide free. In terms of country wise, India, there is a program going on. We using CRISPR Cas9 to make the rice drought resistant. But you can see in different different countries, there is a lot of different traits using CRISPR and talents, which is a gene editing tool, to improve the crops, which is for food safety. So this work was already published earlier, and it is now FDA approved. That they knocked out using CRISPR the enzyme which encodes polyphenol oxidase, which causes browning in the mushrooms. And by delete, so it's a family of genes, and there are 6 genes. They deleted just one, and they reduced the enzyme activity by 30%, and the mushrooms do not turn brown as soon as it is cut. And this technology is now being currently extended to banana and apple as well, so that they won't turn black. The other application that has been published, not in field trial yet, is to engineer wheat. Now we know that wheat has gluten, and there are many people who suffer from celiac disease, which is an autoimmune disorder because there are small peptides in the wheat that cannot be digested by any protease in our system. So they become immunogenic, and that's a huge problem. People have almost 7% of the Western population, and in India, almost 1 to 2% of the population, especially in North India, with wheat eating population, has this disorder. So what was done by this group was to mutate this domain in this gliadin. So gliadin is the one which causes the problem. The CRISPR edited it as so that the immunoreactivity of this wheat is now reduced by 85%, so that the bread or any other product that is being now made using this transgenic wheat or edited wheat will be much safer. Otherwise, those patients, they have to take other cereals which do not have gluten. CRISPR has also been used to increase the productivity of maize in drought conditions. So it is already known from the previous studies that there is a gene which is called ARGO S8, but this is not expressed very highly. But this gene expression improves grain yield under drought stress conditions. So what people have done is that they have knocked in a highly constitutive promoter, a strong constitutive promoter, right next to this gene, so that now this gene is expressed much more. So either they have knocked in or they have swapped the native promoter with this other maize promoter, and that has led to an increased grain yield under drought condition. The uh, the other example that has been done some time back is to increase the rice productivity, and this was a kind of serendipity discovery. They were using CRISPR to knock down abscisic acid control this sensing receptors, and they found that if they knock out certain receptors which can sense this phytohormone, then you can see the grains are much more compared to the wild type. So wild type is in the middle. Here it is wild type, but you can see this knocked out. These plants have much higher productivity and these kernels. So this is another example where CRISPR is used to increase the crop yield. So what are the benefits of CRISPR in agriculture? Of course, as we saw through different examples, that it can lead to better farming productivity, that is more grain, more yield, and better production for per square area. It can also help in food security, because then we can make insect resistant, pest resistant, and even drought resistant plants and crops. It can also improve health because more nutritious fruits and vegetables can be created, which can express carotenes or other important metabolites. Also, we can reduce the allergies caused by certain food by knocking out the genes, as we saw for wheat. And of course, taken together, this will lead to more sustainable practices, because we don't have to make GM crop, but this is not exactly GM. You are knocking out the endogenous genes, and so these will have less of a regulation. So taken together, this technology will in future improve the agronomic traits of different crops which are used for food as well as cash crops, and leading to less pesticide use, less fertilizer use, and maybe less water use if they are more drought and salt resistant. So finally, we saw different examples. What are the challenges? Of course, everything is not rosy and nice. This technology is relatively new, although it is easy to do. But there are certain concerns, and there is much needs to be improved. So one is to which people have seen, there are sometimes there are off-target effects. That means this CRISPR-Cas9 complex can go and edit or do something elsewhere because it is only 20 nucleotide, and the chance of this finding 20 nucleotide in a 3 billion basis human genome is pretty high. So there might be off target effects. Delivery system remains a challenge. These Cas proteins are very big. So currently there is research going on to find smaller Cas proteins. So they can be packaged in viruses or nanoparticles and delivered directly. The integration of the DNA target remains unpredictable little bit. And of course, the how to pack the Cas proteins into different cargo remains a challenge. The modification rate currently is low in humans and plants because the homologous recombination rate is not very high, unlike yeast, as Poonam Madam was saying. So, other organisms have very low homologous recombination rate. So, that needs to be improved using the CRISPR Cas9. Okay, so with that I'll end. So this is the institute where I work with, and we have a teaching program. So any of you or all of you are welcome to visit anytime. We do run workshops sometimes, so keep an eye. We'll have it maybe in this summer when the schools and the colleges are shut. On CRISPR and a variety of molecular biology techniques. My lab is funded from different funding agencies. And with that, I'll thank you